For many years, there were many discoveries in basic science and molecular neurobiology, identifying molecules that potentially could be important for mental health or mental diseases. Now, the application of all this knowledge to the human condition is still elusive.
We don't associate a disease with a single gene. It's usually across many different levels of gene association with a particular disease. It requires computational models to try and understand the relationship between genes and particular diseases. This is ultimately a mathematical computational challenge, a statistical challenge. The problem of understanding the brain was much bigger than any one of us could handle, and that was the original premise of the Ludmer Centre.
The Ludmer Centre has been an initiative between McGill, the Douglas Institute, the Neuro, and the Lady Davis Institute. It began initially bringing together scientists interested in brain imaging and in epigenetics, as well as analytical methods, and has evolved over time.
The Ludmer Centre is a nexus for people with different scientific backgrounds to come together to understand how the brain works. The motivation was to create a center for big data applications in neuroscience. On top of that, it provides a training environment for the young students coming through to learn about multimodal big data approaches to science. We develop techniques to analyze the data, develop computational models of how the brain works and how it breaks down in disease.
Part of the mission of the Ludmer Centre is really to integrate different types of data and use these resources that are available to different researchers to make sure that we cover either the life course or different modalities to help us understand the mechanisms and the associations with mental health. The Douglas has a long tradition of studying how the environment influences gene expression and health and disease. We can do more molecular, more basic science, but we also have the clinical setting. This back and forth between basic science and clinical science takes many, many years. But the environment that we have here favors this interaction and accelerates discoveries. Through the Ludmer, we have collaboration with Dr. Celia Greenwood. Dr. Greenwood is a statistical geneticist, and she is the director of the statistics genetics part of the Ludmer Centre. And her expertise in stats genetics is important to try to disentangle these questions and understand mental health more broadly.
There has been an opportunity to expand the Ludmer Centre into the single-cell genomics. It's a way to actually interrogate every cell at a high throughput manner. You're able to basically understand what's going on at the level of the cell in a tissue all at once.
We can go with a higher resolution and create cell-specific expression-based polygenic scores, which is amazing in terms of exploring more detail the mechanisms associated with the conditions being investigated. We can use it to inform the calculation of polygenic scores very soon.
The Single-cell Transcriptomics Initiative is going to deliver ultimately 9 new faculty distributed across 3 different institutions involved in the Ludmer Centre. So that just by itself is transformative. Now, it's all very well to talk in these grandiose terms, but you have to have the informatics infrastructure to handle all of this. How do you make it available in such a way that it can then be mined to find the associations between changes in gene expression of specific cell populations? How does that relate to disease? So there's a whole new frontier that we have to build for the Single-cell Transcriptomics Initiative. One of the things that has changed in science over recent decades is the rise of open science, where researchers from all over the world are willing to share their data with other researchers in a completely open manner, so that more can be gained by the society in general. The Ludmer Centre has been a great proponent of the idea of open science. This big brain data set has been downloaded 25,000 times. People are making use of this data in ways that we hadn't thought of.
There's a lot that can be done to improve accessibility and making sure that the scientists have the support that they need to share their data. The team needs to support the staff that is involved in collection and in curation of the data.
The Douglas Brain Bank collects brain tissue primarily from the larger Montreal region, from individuals who died from psychiatric illnesses, car accidents, or died by a heart attack. We aim to have brains from all age groups, socioeconomic classes, from all demographic variables, to be as representative as possible. The Ludmer Centre does not support directly the brain bank. The Douglas Institute subscribes to open science. We are working to build a platform that will connect the brain bank and the data that's produced to a database that will be open to the scientific communities.
We benefit tremendously from the community that is involved in the Ludmer Centre. The Ludmer Centre provides this environment of collaboration and interaction, not only for the scientists, but also for the students.
Science is not eureka moments. It's usually the product of decades of hard slog, incremental advances, and putting an infrastructure in place which allows all of that to happen. Irving understood that from day one. This has allowed the Ludmer Centre to flourish because you have a donor who gets it. It's a meaningful enterprise because it's not just doing science and publishing papers, but it also has a global impact on brain health.
We are all very ambitious, and we want to really understand what happens in the brain in terms of functioning. We want to as well develop interventions, treatments, but just by advancing knowledge and understanding how the brain works, to me, it's a big step.
Good morning, everyone. Bienvenue, bonjour à tous et à je m'appelle Lesley Fellows. I'm Lesley Fellows. I'm the Vice Principal Health Affairs and the Dean of the Faculty of Medicine and Health Sciences in, I think, day 62 or something like that of my mandate. I will say that some people are counting down how many days are left for me, but it's really a privilege for me to be a host and moderator. Welcome to the Faculty of Medicine and Health Sciences. Welcome to the beautiful building here where our administrative teams are situated. It's a particular pleasure for me to welcome you to this session and to be part of this session because I'm myself a neuroscientist and quite interested in many of the topics that the Ludmer Centre is tackling, so I'm really looking forward to the exchange. And thank you for being here and homecoming and for staying connected to McGill. Before I go further, I just want to acknowledge that McGill University is located on land that has long served as a site of meeting and exchange amongst Indigenous peoples, including the Haudenosaunee and the Anishinaabe peoples. McGill acknowledges and thanks the diverse Indigenous peoples whose presence has enriched this territory on which peoples of the world now gather. And I feel that acknowledgement's worth reflecting on just for a moment because it causes us, I guess, to think about the complicated history of our interaction with the Indigenous peoples. And these days, certainly in the Faculty of Medicine and Health Sciences, our efforts to really look to ways to work really constructively and effectively and inclusively together with the Indigenous peoples. And I feel that's a broader message these days that we need to find ways to to connect and move past difficult histories in the spirit of reconciliation. So without further ado, let me move to the Ludmer Centre. McGill's been around for 200 years, the Ludmer Centre since 2013. A very important addition to our ecosystem. It's called the Ludmer Centre for Neuroinformatics and Mental Health. It's a McGill-affiliated research center that's goal is to drive innovative, collaborative, and global big data research. The idea is to build a core infrastructure, as you were hearing in the video, that's needed to strengthen and reinforce transdisciplinary partnerships. People talk about transdisciplinarity, but when you come to doing it, you realize how many gaps there are between the disciplines and how much very practical work is needed to create the space and the tools and the vision to move to working together across even very nearby disciplines are still far apart in many ways. So I'm excited that the center's at McGill. It's very visionary in its efforts to connect our neuroscience strengths at different McGill-affiliated sites, the Lady Davis Institute, the Neuro, and the Douglas. The list of core research themes interests me because looking at them, you might think it's not obvious how they connect. And we're going to hear this morning how they do. The themes run from functional genomics and epigenetics. And if you're not familiar with those things, you're going to hear more momentarily about them. But also neuroimaging and biostatistics. And now, most recently, single-cell genomics and the neuroinformatics you need to deal with that really complex data. So thanks to a donation from the Irving Ludmer Family Foundation, a recent donation, not only the center, but the effort to move into single-cell genomics is being supported in a really interesting way. And I won't explain to you what it is. I'll let the experts tell you more. But the idea is to look at individual brain cells, cell by cell, and try to understand, which is a daunting prospect, try to understand how these cells interact, to eventually get to the point of understanding brain diseases and normal brain. I'd like to welcome and thank our founding donors, Mr. Irving Ludmer and Ms. Freeman Lander, who are sitting here in front. It's great that you're here with us today. It's their generosity that led to the creation of the Ludmer Centre in 2013 and a new donation in 2022 that's allowed us to do the single-cell genomics project that's well underway. And without further ado, I'm going to introduce our speakers. We have two this morning. Professor Corina Nagy, who's a researcher at the Douglas Research Centre, an assistant professor in the Department of Psychiatry at McGill, of course. She's involved with the Réseau Québécois sur le suicide, les troubles de l'humeur et les troubles associés. That's the Quebec Network for Suicide, Mood Disorders, and Related Disorders, and is co-director of the Douglas Bell Canada Brain Bank that you were hearing about in the video. And she's a principal investigator at the Ludmer Centre. So maybe I'll stop there and let you have the floor and hear about your work, and then we'll have Dr. Silveira tell us what she's working on, and then a discussion. I'm hoping to make this very interactive, so stay awake in the back.
So I think my biggest challenge is going to keep to 12 minutes. So you guys can calm me down if I keep going. Set a timer. So anyways, thank you very much, Lesley, for that lovely introduction. I'm super happy to be here to sort of explain how single-cell technology, I guess I should switch slides. There we go. How single-cell technology has enormous potential in neurobiology in both health and disease. So I want to start with a little analogy to put this technology into context. I'm going to start with the world. This is a very vast geographical landscape that has a lot of physical differences that influence us as humans in terms of our culture, our values, and that influences how we interact with each other. And so, we might interact very differently even within North America. Even if we start to look a little more homogeneous, we still have a lot of differences that may not be fully recognized. And we might distill that down to some stereotypes. For example, we might say that Canadians are better at dealing with cold weather and snow, and perhaps we're overly apologetic. And maybe we don't say the same thing about Americans. We can also say that within Canada, we have very different cultures and very different values, and that people in Quebec may interact differently with their milieu, and across Canada. They may interact differently with people in Alberta. And so what I'm trying to get at here is that we can go down and down into further resolution and understand that the regions that we're in, the contexts that we're in, even with this group here, McGill, kind of define our values, and those values define how we interact. And so here in this milieu, in this scientific milieu with my peers, I will act a certain way, and I will talk to you about science. But if you take me out of this milieu and I go home and I interact with my children, it's going to be quite different. And I want you to take this concept and sort of apply it to the brain, because that's kind of how it works as well. The brain is a very large organ. It's broken down into multiple regions that have very specific functions. We have frontal, parietal, occipital, and temporal, and these have, functions that we've defined for a long time. But even within those regions, we have subregions that are defined by Brodmann, for example, and they have very specific cytoarchitectures. And within those regions, we have even more regions. And these are your layers, your cortical layers. And within those cortical layers, we have individual cells. And those individual cells will interact very differently within their cortical layers, and across cortical layers, and across regions. And all of these interactions are defined by their gene programs. So what is a gene program? Well, that is the set of transcripts that are being expressed in any given cell at any given time. And prior to this, all we could do is know that there's a great diversity of cells in our brain, but what we would do is take our brain and take a region and homogenize it down and get a very general signal. Now, a general signal isn't necessarily wrong, but it's also not particularly accurate. So it's kind of saying that, well, we're not Canadian unless we're super apologetic. And of course, that sort of neglects all the other features that we have as a population. And that's neglecting, it's the same concept with cells. If we just take the homogeneous signal from the cells, we're neglecting a lot of features that are important to their interactions. So these gene programs are really something that we can now define. We can go in and understand which genes are being expressed, and as a function of the expression of those genes, we can understand features of the cells. So we can understand their morphology, we can understand their function, because, for example, the genes lead to the generation of certain proteins. Those proteins will become receptors and other molecules that allow cells to interact. And they will also influence the electroconductivity of a cell. So all of these things can be really defined by their gene programs. And we use ultra-resolution look at these individual gene programs and understand these morphologies and functions, but we can go back in and with quite good accuracy actually label where these cells are coming from in the brain. We can say, you know what, this gene program says that this cell is coming from this particular layer in the cortex. This is quite powerful. And we can also know, for example, as we go back to our kind of analogy of the world, we know that the neurons in the visual cortex are not doing the same thing as the neurons in the prefrontal cortex. And so we have now this refinement. And I hope that you can understand the power this gives us for looking at diseases. So, for example, in the context of neurodegenerative disease, perhaps this makes us aware of which dopaminergic cell specifically in the substantia nigra first to die. Because as we know, Parkinson's results in the loss of dopaminergic cells, but there's a very specific pattern. So if we can define these patterns by these functional transcriptomic gene programs, then we can know more about why those particular cells are dying off, and when and how. We can look into the clonality of cancer cells. We can look into exactly which cells have an infection, for example, which have been infected by viruses, because perhaps not all cells are. We can use this in the context of drug screening and drug development, where we can take a molecule and we can see how those molecules are influencing the gene transcription of a particular cell type and where it might be more relevant. We can also use this for biomarker research. Maybe and gene programs better, we'll be able to actually have some indication of prodromal information so that we can get at a disease before it progresses into its symptomatic states. So here's some real-life examples of the data that we've generated using this technology. What we've done here is we used the brains of individuals who had histories of depression and controls. Those are people, as explained in the video, whose brains were collected by the brain bank but had no pathology. And in this particular study, we used only males. And what we did is we took a very specific region, Brodmann area 9, if that means anything to anybody, and we broke up the tissue into its elemental components. And the elemental component, of course, is the cell. And we then sequenced every single one of these cells to generate their transcriptome. And based on their transcriptome, their gene profile, we're able to actually cluster like cells together. And all those cells that are more like other cells will cluster together in this t-SNE map. And those that are a little bit different will cluster away. And based on that, we can actually annotate all these cells in the brain to the resolution of their cellular cortical layer. And so in doing this, we then took all the cells that belonged to individuals with a history of depression and compared them to controls to see how their gene expression levels were changing. And we found that actually quite a bit of it had changed. And most of those changes were actually accumulating in 2 specific cell types, in deep layer excitatory cells and OPCs, which is an oligodendrocyte precursor cell. This is an immature cell that isn't actually that abundant in the brain. With bulk analyses of brain tissue, we would never have gotten to this resolution. We would not have been able to pull out, and in fact, these signals had not been pulled out previously in research on depression. So this signal in the OPC is something that we were able to pull out using this technology, and the specificity of the neurons that are implicated also was pulled out because we can use this technology. And so now we can make inferences about how these 2 cell types might be interacting and perhaps how OPCs might be modulating these deep layer neurons. But I just said that we did this only in males, and this is a big problem in biomedical research in general, because females have often been overlooked. We basically try to take one sort of sample and generalize it across samples, and that just really isn't enough, because females and males manifest diseases differently. In most neurological neuropsychiatric diseases, we have a skew to one sex or the other, and in depression, well, it's twice more prevalent in females than it is in males. And also, their symptoms manifest differently. So it's not sufficient to just look in males and say, this is what it's going to be like in a female. So, of course, we had to get at this question. So we generated more data. And so in our first study, we generated 80,000 cells. We generated the transcriptome for 80,000 cells. We doubled that. We more than doubled it. We doubled the number of samples, but we more than doubled the number of cells, and we now are looking at 170,000 cells. And so we were able to basically pull out the same signals. So the first thing we did, of course, is make a unified pipeline so we could look at these data together. And then we integrated, with this integrated data set, we were able to look at where the most abundant signals were coming from. We were able to recapitulate this finding in males, in this deep layer excitatory cell type in OPCs. But in females, we saw that the signal was actually coming from a different cell type, a very different cell type. It was coming largely from microglia. So the microglia seem to have this important influence on the pathophysiology of major depression in females, which we just weren't seeing in the males. And this really speaks to how we need to have this resolution when we're studying pathologies. Now, I don't want you to walk away from this talk saying, okay, males and females are completely different and that, we can't look at them at all together. This is not true. So these are RRHO plots, a little bit weird to look at, but basically the bottom left and top right corner suggest anything that's concordant. So when we looked at differential expression in each individual cell type, the things that were most salient, the things that crossed the threshold, those would be extremely different. But if we just rank all the gene expression changes and we look at, okay, what's increased in males in cases and controls, so what's increased in males with depression and what's increased in females in depression in these specific cell types. By and large, these were concordant. So by and large, our brains are working more or less in the same way. But those that are really, really saliently different, they are in a different, have a different pathway, they represent different cell type and distinct genes. So, you know, our ongoing hypothesis for this is perhaps there's a different start point, and that's where we need to kind of examine this more. Perhaps where the baseline in females is a little bit different than the baseline in males, and so the trajectory that we need to take, or the trajectory that is taken, we don't necessarily need to take this, to end in depression is different. So where do we go from here? What is it that we are trying to do on top of just understanding the pathogenesis and the physiology of depression in the brain? Well, of course we want to understand how we can better prescribe existing drugs or even develop new drugs. And so our typical developmental pipeline for drugs looks like this, or at least prescription pipeline is that you go see your GP, you say that you have a pathology, they kind of prescribe the most popular drug. And in the case of depression, well, this works in 50% of people. And it will also not work in a large number of the population and also have very adverse side effects in another set of the population. And this framework can be sort of applied to many disease states. What we need to do is have some sort of indication, some sort of insight into the individual so that the molecule we're prescribing is more suited to their case. And that way we're more likely to have positive outcomes. And so using this type of technology can get at those answers, perhaps. So it would kind of look like this. What we've been doing before is we have this homogenative cells. We get this kind of generalized signal. We use a molecule toward that signal, but it's not enough. It lacks the precision. In some cases it works. In other cases it doesn't, because in other cases this is just not the profile. And what we need to be doing, if we kind of take this example of the males and the females and what we've just found, perhaps what we need to do is actually develop molecules that will target this subtype of cell, and particularly perhaps those states. So we have a number of molecules that would be suitable for those particular, for that particular phenotype, if you will. And then perhaps we'll get a much better, more efficient and precise outcome that is favorable to more people. So the last thing I want to touch on, which is a really important mandate, as was explained in the video, is making all of this information open. Because what I just described to you is 170,000 transcriptomes. That's what I just described. That's an incredible amount of data. And that incredible amount of data will do nothing sitting on my hard drive or in an external, in a locker somewhere. So what we've done, and what we plan to do moving forward and integrating with the framework that's already in place, is to make this data available as such in an app that is accessible. So if you guys wanted, so you've seen that I've dropped a bunch of QR codes throughout this presentation, so you can look at these particular papers if you like. You can get more into the science, because I kind of went very high level. And you can also look at this particular app. You can go in, you can put your gene of interest in there, you can see which cluster that gene is most expressed in. So if your favorite gene, I threw out ApoE here, if your favorite gene is something else, you can see exactly which clusters, which cell types these are expressed in. And you can get all kinds of different information. We've done this really to try to engage the larger scientific community, because alone we will approach a problem from the same perspective. Together we'll approach it from many. So I just want to say thank you. I want to thank this group of individuals I work with on the daily. We clearly cluster together. I want to thank the funders who believe in me, and of course an enormous thank you to funders like Mr. Ludmer, who have enabled advancement in science that would otherwise not be possible. So thank you very much.
Thank you, Corina. I think we'll hold questions for the discussion at the end, if that's okay, just in the interest of kind of time and coherence. So we're going to now get sort of a second perspective on this from Dr. Patricia Pelufo Silveira. Dr. Silveira, I know a bit better than Dr. Nagy personally, and I happen to know that she's made her journey to McGill from Brazil and is just another example of McGill University attracting outstanding researchers and clinicians from around the world. She's an associate professor in the Department of Psychiatry, associate director of McGill's Integrated Program in Neuroscience, the largest neuroscience graduate program in North America. She's a researcher who works on environmental adversity, neurodevelopment, mental health at the Douglas Mental Health University Institute. And she's the scientific director of the Ludmer Centre for Neuroinformatics and Mental Health. So she's going to give us a perspective on the same sort of problem, but from her point of view, and then we'll be able to kind of dig into this kind of cutting-edge work and talk together. So at the end, we all walk out with enough of an understanding to discuss this at the cocktail party later tonight.
Thank you so much. Je suis tellement contente d'être ici aujourd'hui. I'm very happy to be here sharing with you the work that we do in my lab. And thank you, Dr. Fellows, for the introduction. I was trained as a pediatrician, and in the clinic, whenever we see a baby like this one, we're happy, right? She has everything that is needed to survive. She's breathing on her own. She's calm and sweet and is ready for a whole future ahead of her. However, when we compare this baby with another one that was born on the same day with the same gestational age, we realize that there is considerable difference in size. And you can easily infer that it's not only about looks. Her smaller external size represents smaller organs. She'll have less beta cells in her pancreas, she'll have less nephrons in her kidney, and this will persist throughout her life course. And this anatomical deficit is, of course, coupled with a functional deficit that will have an impact on health and development and health and disease trajectory throughout the life course of this child. And how prevalent is that? Well, in May, one of the best medical journals that we have, The Lancet, released a special issue calling our attention to the high prevalence of children born either too small or too early. And if you put these two categories together, these small, vulnerable babies represent one-fourth of all births worldwide. This is true everywhere in the world. It's independent of the economic status, the political views, the hemisphere, and I want to emphasize this. This is true everywhere. And in Canada, the prevalence of being born small is actually increasing in the recent years. So the causes of low birth weight go way beyond nutritional causes. For example, diabetes increases the low birth weight incidence, advanced maternal age, obesity, chronic stress, hypertension. But the impact on child development and their health disease trajectory is similar. Imagine 25% of our population is affected by this. My lab has dedicated its existence to understanding how these events that happen very early during development, even during fetal life, impact the child's brain development, behaviors, contributing to the development of these diseases in the long term. And especially, we've been investigating the higher level cognitive skills that these children use to control and coordinate behaviors and other cognitive abilities. For example, making decisions about which food they're going to eat, healthier, less healthy, how much they value the rewards from the environment, how they behave towards environmental cues, their capacity to resist or refrain from temptations. And of course, if we talk about behavior, our focus of study is the brain. You see here the synapse, which is the point of communication between two neurons in the brain. And this structure is important because here is where the information is transmitted from one neuron to the other. And ultimately, this structure is responsible for how our actions and our behaviors end up being expressed. So the synapse is a complex structure with multiple proteins and enzymes working in this coordinated effort to reach this common goal of transmitting information between one cell to the other. Of course, we cannot really get into the detail of this process in a living child, so we have to establish methods that infer how children vary in the way that this structure works. So my lab approaches this issue by employing multi-level data integration to dissect out why some children have, for example, better attention skills than others by using the triangulation of multiple data sets, combining genomics, neuroimaging, and other approaches. Now, most of the studies so far have done this analysis by investigating each one of these molecules that you see there, one at a time. So you run these large studies and eventually identify that this molecule here seems to be important in a certain condition. And then you dig deeper and try to understand how it works and why it is important. However, this approach ignores the fact that all these molecules here are working together in a coordinated effort in this beautiful dance that is the synapse. So my lab has created a methodology to start considering all these different components together in a same marker. So instead of focusing in a single gene or a single protein, we focus first in identifying how these molecules are working together in this biological process, forming this network that you see here in the figure. And after that, and through a pipeline, a sophisticated mathematical pipeline, we're able to characterize variations in the expression of this network in different children by using their genomics information. In other words, we are able to identify how the children vary in the way that these genes are working together in the specific brain regions of their brain. And at the end, we can have a score for each child or even each participant in a sample that helps us have a picture or a measure reflecting the super discrete biological processes that are hidden there in the molecular intricacies of their brains. And we can do that for a large number of individuals and study how variations in biological processes influence health and disease over the life course in populations, large populations. We have published a number of papers using this technique called the expression-based polygenic scores. I put a QR code there where you find these publications if you're interested in learning more. And I'll give you an example on the application of this technology. In this work, we are interested in identifying children that would be more or less vulnerable to the long-term effects of neonatal adversity that is plotted here in the x-axis on cognitive flexibility at 4 years of age, so 4 years after birth. Neonatal adversity in this study was defined based on oxygenation at birth, and it increases towards the right. So the more you go to the right, the worse was the levels of oxygen at birth. And in the y-axis, we have cognitive performance in a task applied at 4 years of age, as I said. And this is right before entering school. And poorer performance increases as the values increases. Sorry. So we see that for the children depicted in blue, in fact, there's no effect of neonatal adversity, and their cognitive performance is overall good. There's no difference depending on the exposure that you have in terms of neonatal adversity. But for the children that are depicted in orange, as you have worse and worse levels of oxygen, you have worse and worse performance 4 years later. And the only difference between the two groups of kids is the score that was created in our lab based in a gene network created from their prefrontal cortex, inferred by other types of data. And this is super important because these children depicted in orange will probably benefit from receiving support, training, specialized care, more personalized education as they enter school, while those that, the blue ones, even those that were exposed to adversity, this is not necessary. So my lab was also able to identify in which parts of the brain these effects were taking place. So you see here some parts of the thalamus and other cortical areas that are important and crucial for attention skills. Now, as Corina said, we are quickly advancing towards an era in which the resolution of gene expression information is improving dramatically. Now, the data that we use for triangulation, that I used to create this course, more and more can be generated from the single-cell technology, so we have the resolution at the cell level, as opposed to the brain region level. This is incredible because we now can identify gene networks that are specific for each cell type. Brain cells are extremely different, as Corina said, so the way that these gene networks are formed and the way that they work in different cell types is very, very different. As a consequence, the way that individuals vary in these gene network function is very particular. This is a very specific signal, and being able to capture variation at this level in children and in patients will decrease the noise that we have in our scores. This refinement of the data will allow the creation of much better scores that can define with more precision which child will benefit from support and interventions and which child can continue their regular trajectory without needing to receive extra support or extra care. Imagine how much this can save in time and resources for the families that really need, and how much we will be able to test and plan tailored supporter strategies specifically for those kids that need. And finally, these more specific networks can also help us identify central molecules that have an influence in the function of the entire network, like the ones you see in darker colors here in the figure. And in the future, this has the potential to lead to the development of better and more targeted pharmacological agents, for example, that will act specifically on those points of the network and influence those molecules in those cells. So the landscape for improving treatment is amazing. It's very and especially through protocols I described to you, the scores, opens up a gigantic venue for investigation with large potential to impact, to guide the creation of preventive and therapeutic, precision medicine, concrete, and especially for fields of medicine that have traditionally been the periphery of these advances, like pediatrics, psychiatry, and also in complex scenarios, complex clinical scenarios. For example, when you have the coexistence between mental disease, psychiatric conditions, and metabolic conditions in the same patients, which usually are very challenging to treat and to assist. So I would like to thank my lab for their dedicated work on this, the funders, and especially the Ludmer Centre for providing a thriving and supportive environment for innovation, and you for your attention. Thank you.
Start with a bit of a let me bounce an analogy off you and get your thoughts about whether this is helpful to understand how you're trying to approach this. I mean, one of the major problems in studying brain disorders is the complexity of those brain disorders and the variability to the point where we don't even necessarily agree on the diagnosis. So even all of the ones that have been discussed today, whether it's depression or neurodevelopmental disorders, that even defining the edges of these conditions in clinical contexts is not easy, which of course makes it hard to understand the underlying biology. So what you're talking about is kind of coming at it from a different way, in a way kind of bottom up, to see where the patterns are, that we know that these are heterogeneous conditions, and can we find patterns there that might help. So I was thinking about opening a newspaper and trying to understand what was going on in that newspaper by analyzing it letter by letter, by looking for all the letter A's and trying to see if there are some kind of pattern to that, or by looking at the drops of ink and how they bind to the fibers of the paper. Obviously, or by looking at it word by word or paragraph by paragraph. And obviously, those different levels are more likely to yield meaning in this example where we know where the meaning comes from. But in the work that you're doing, we don't actually know where the meaning comes from. So how do we know what level to target? And do you worry at all that you might be studying the drops of ink when in fact we need to be studying the words, or that we're studying the words but the problem is at the level of the drops of ink and the words will never give us the answer? How do you navigate the level of resolution to study?
Do you want to start, or do you want me to start? Okay. So I agree with you 100%. It's this idea of how reductionist can you be about such a complex disorder that, is way more than just your brain, right? It's also your interactions on the everyday. It's all the experiences that you've had. So, I mean, it's a question that, as a scientist, you have to reflect on all the time as well. Am I approaching this in the most absurd way possible? Am I really going to get something out of these drops of ink? But, the way I see it is we do need to have a baseline. So we'll start with the drops of ink until they make a letter. And then we'll get to that letter until they make a word. And then we'll get to the word until they make a sentence and a paragraph. So it is going to be, I think it's not going to be, it's faster process because we already have some of the context for those letters and those words, and we can put them into those contexts of all the early neuroscience research. But it is there. That is what it is starting at, but we do need this baseline. So then we can understand how all of these, other sort of features are influencing the way that the words are written and if they're misspelt. And that's really the way I have to look at it, because otherwise, I will get sort of bogged down in this idea that it's too specific or whatever. But this is what we're doing. We're generating the baseline and we're going from there.
Yeah, I think that's a perfect answer, Corina. But also, we need to think about more and more today, the way that we address these challenges and deal with this amount of data is automatized. And we have softwares that help us, like Corina showed at the end of her presentation, softwares that help us understand patterns and make connections and understand how one thing connects to the other and the different levels of complexity. And I think this is one of the benefits of being embedded in the community of the Ludmer Centre, because we have experts that are part of the community that develop these softwares and know how to use the softwares very well and extract information from different complex layers, put them together in meaningful messages. So today, I don't think we work anymore in the classical, the caricature of a scientist thinking alone in a room and making these connections myself in my office there. We actually have a huge team of people, data scientists, mathematicians, statisticians, people from different backgrounds that actually help us make sense of these different intricacies and complexities.
Thank you. We'll start at the back, the green jacket.
This is a question for Dr. Nagy. When you were talking about the implications of the single-cell genomic research to be used for individualized pharmacology treatment, I'm assuming right now all of these sequencing that was done are in post-mortem brain from the brain bank. Are we at the point where we can do that in vivo, and if not, how far are we? How will we even get there?
That's a great question. So, typically we won't use, tissue from postmortem samples to do drug screens because they're not alive anymore. But we at least know what cell types we want to target. And so then we can get those cell types from primary cultures or immortalized cultures, and we can actually test different molecules to see how those molecules are influencing that particular cell type. So the data that we generate from postmortem tissue is the starting point, and then we need to kind of expand that out into in vitro and in vivo models that do include animal models. Although I do want to put a caveat out there, which is kind of exciting from a perspective of somebody who studies, human primary tissue, which is that animal models are amazing, and we need to use them and leverage them, but this single-cell technology has shown us that not all the cell types are actually the same between species. And we have cells that are unique in our brain that just don't exist in rodents. So that was just kind of a really exciting kind of data that we got from this work.
But then for therapeutic uses, how will we take cells from a person who's still alive?
So we won't. Okay, we'll just test how those cells are reacting to the molecule, or we can actually treat those cells in a way. So we have different compounds that we can use to kind of make the cells work or miswork, if you will. So we can use different compounds that will stop them from firing or induce inflammation. And we can make that environment for the cell and see how it responds to the drug. But typically what we want to do is really just test how the cell is responding to the particular molecule, and then we can have a better idea of whether or not it's responding in the way we want. And if we apply some of Patricia's data, for example, where she's looking at very specific mechanisms within the cell, then we can actually target those particular proteins, for example, within that cell and see if we're getting what we want.
Yeah, just I think that the data that Corina generates can help us develop markers, like the ones that I showed you in my talk today. And this we can collect from, in my lab we collect buccal swabs, and they're super simple to, painless, very easy to handle. And this is enough for us to have, using her data to apply to other tissues and that are accessible in large populations.
Okay, here I think, and there's lots of questions. We'll get to you all.
Thank you very much. Just as a continuation of what Dr. Fellows was asking. So, I mean, nowadays we know that AI has learned to talk to us like through ChatGPT, going through those letters and corpuses that were built. So we fed them, when we had enough books, we fed them to AI, and now AI can talk to us. So hopefully at one point, the data that you're generating would get there as well, and definitely it needs numbers and size. But what I want to add to that is two things that you mentioned. One of them is the time and the changes. So I think that we are not dealing with a newspaper. We are dealing with a interactive tablet that the words are changing. Mm-hmm. And how are we addressing that? And another thing that I wanted to know is that why McGill, and what do we have as a core competence to contribute to generating those corpuses?
So to answer your question about the ever-changing gene profiles that we're dealing with, I mean, you're absolutely right. And I think it's really interesting because we can't just look at static time points. We have to actually integrate information from all kinds of time points. And that's some of the work that we're doing now. We're trying to see how cells change their sort of transcriptomic profiles through time. And we can do that with models. We can do that with induced pluripotent stem cells. We can do that with a bunch of different mechanisms. And we have to basically generate those data so that we can understand a temporal aspect to this gene program that we've been talking about, and then feed those to the AI. So basically, of course, we need to generate still much, much more data to truly understand what's going on and to be aware that these are not just, stagnant snapshots of what's happening, but we need to be aware that our brain is continuously evolving and changing, and our cells actually toggle between states. So, there's a bit of controversy in this field right now because we try to annotate the cells the best we can, but sometimes we're not sure if we're annotating a cell type or a cell state. And so we're kind of reluctant, so I kind of use very general terms when I did this presentation, but we're a little bit reluctant to call all these dots in the t-SNE a cell type. We typically actually call them clusters that represent an overall cell type that might be in a certain cell state. And I think that, that's really important to acknowledge and be aware of. But I think it's also part of the information that we need to use moving forward. So why McGill? One might argue, why not? But also because we've been in this game really since the start, right? We started when the single-cell technologies came out. We have an expertise in it, but we also have amazing resources like the Brain Bank, like the framework that's set up at the MNI for testing drugs like the EDDU. We have a very nice neuroinformatic platform that was set up. And so I think we have the infrastructure, and with really nice donations to actually engage and bring in more talent, then we simply have everything we need for this recipe to work. I'm sure you have something to add.
And we have, as you said, we have the tradition in neuroscience. McGill is a leader in neuroscience for decades, years, and we have so much talent. And I think that we are profiting from this history as well.
Absolutely. Thank you. I think here, and then after that.
Okay, so I have two questions addressed to either doctor. First of all, I have a sister who's just recently turned 77 years old. One of the topics you touched on was depression. So she's right now in a psychiatric hospital, the Hippolyte Lafontaine Hospital, getting electroshocks. Is that likely to help her snap out of depression, or is this just hocus pocus? The second question is, and I don't want Elon Musk getting jealous of me, but I'm 75 years old and I have a 27-year-old girlfriend in Ukraine who graduated in the spring of 2022 in pediatric medicine from Odessa National Medical University. So if she ever makes it to Canada and we're able to get married and we procreate, she'd be in her late 20s to early 30s. I would be in my late 70s. I'm 75 now. You talked about one of the causes of defects is older mothers. She would be in a reasonably good age range for procreation. Is there any adverse effects having an older father, like in late 70s? As I said, don't tell Elon Musk. I don't want him asking me dating advice, and yeah.
Sure, I don't know what the qualifications are here, but also personal health advice is not the main purpose of this interaction. But go ahead.
No,
yeah.
I'll give my answer after.
Okay, you can always take it to the hallway for personal advice about
Yes.
about toddlers at the age of 75. Do you want to answer the first part of his question about depression and the treatments for depression? Is that something either person feels they can answer?
If you
So, I mean, if we look at the research, to answer your first question about ECT, if we look at the research, this is still one of the main treatments for treatment-resistant depression. And actually it's been studied quite extensively, and it does have almost a reprogramming effect, if you will. So this is stimulating your cells in ways that they don't typically get stimulated. And it would be, if we had this capability to look at the cells at a single cell level after this, I mean, we do this. We do, kind of give these types of therapies in vitro to see what's happening. And we do see that there's an incredible change in gene expression. And so this actually tends to be an extremely successful treatment. So yes, there's hope. And but of course, this is not your first line, because it's a difficult treatment. So this is why it tends to be a second line treatment, like a lot of the other novel therapies are second line. So that's to answer that.
Maybe just a word about this type of therapy, because it evolved tremendously over the time, and today this is something that is done under anesthesia, and it's very it's a very standard treatment. So it's very different from what we may think of in movies and all that. So.
Please, thank you. Go ahead, in the front here.
Thank you. Just to give you a little bit of background of who I am, I'm a computer and electrical engineer and a family physician in pediatrics. So my first question to Dr. Silveira is, the data that you used, was it just on the 1/4 of the kids who have issues at birth? or are using all the data? My second question will be about prevention. So,
Yes, no, we use in all kids. So you remember the plot that we had seeing axis. So we have kids that were not exposed,
So
close
the
to
blue
zero,
line, yeah, that was the
up
blue
to
line.
those that were super exposed. And this is actually a population that is not at risk, is a community-based sample. So it's really kids that you don't see a huge difference. They're not super risky. It's a subtle difference in our daily life, but you still can detect such a huge difference in terms of performance. So, yeah.
So it's interesting because in pediatrics, so I've been working for 24 years, and my logical brain from 0 to 18, we don't really see that difference. We don't see, it's not based on prematurity, how young they were when they were born, what size they were, but we can detect very early that the child is going to have issues. By 2 months old, I can already tell you that this child will have an issue. And yet, nobody comes to see us in the office to say, this is what we should start doing. So I'm very interested, I guess the opposite, it's like you were saying, what can I do at 2 months to start telling the parents, this child is going to have anxiety, mood issues, attention issues, all kinds of things. But instead I have to wait. And so at 2 years old, now it's official already that they have issues. And then there are no services at all, zero. It's just getting worse and worse. So you're saying we're going to target services, but by the time we already know who's going to need the services. So I can't even take away from, there's nobody to take away from. There's just people to give it to. So what am I supposed to do at 3 months? So I'm waiting. 3 years old, they're put on a wait list. At 5 years old, they're kicked off the wait list. Now they're in school. And 5 years old, they're having issues in school. It's too late.
Yeah, thank you so much for raising that. And I hear you. I worked as a pediatrician too and facing exactly the same problems. Unfortunately, this is the reality. This is much more about the system than about biology or genes, but it's very true. And if we can even expand what you just said to the prenatal care too, right? The more we prevent during pregnancy and before conception, the less we'll have scenarios like that, and is a huge potential for saving resources for those who really need. So I completely agree with you. And
but I think there's a non-pharmacological work to do and interventions. And is anybody working on that in order to help us?
Yeah, there is work, and I'm part of the large network of scientists based in the States, but we're expanding to Canada too, working very close to communities and pediatricians. So we created a network of putting these three forces together, scientists, pediatricians, and the community, to try to solve these issues exactly about access and early identification and providing services. There's a lot to do, of course, but yes, there is people that is thinking about this.
Part of that? Can we join that?
For sure, yes. Just reach out. Thank you.
One here and then at the back. Yeah.
Yep. Hi, I'm Christine. I did mechanical engineering here at McGill, here for the 40 years. I think the initiative is phenomenal. I've learned lots, didn't know a lot about it. I like the open science aspect. If I understood correctly, you have a brain bank. Are you going to be inviting other, maybe not immediately down the road, but are you going to be extending an invitation to other institutions that have their own brain banks, such as ALS, or maybe some other places, because they're doing their own independent things, and it would be pretty cool to be able to have some of that in there and to connect things, especially with the big data aspect of it. Any comments on that?
Yeah, absolutely. So I talked about this aspect of the brain bank, but the Douglas Bell Canada Brain Bank has over 3,500 brains, and they're from all these different pathologies. We even collect the ALS brains. So there are researchers currently working on these things, and the Ludmer Centre includes some of those researchers, but also we at the Douglas have a hub so that we can sort of democratize this, this technology, so that everybody can actually use it toward whatever question they have. So certainly it's ongoing for almost all neurological and neuropsychiatric diseases and other parts of the body as well. So it's not just the brain that benefits from single-cell technology. So And there are other banks, but we are the most open and largest in Canada. We are a network. We are in constant contact with all these other brain banks throughout Canada and the US.
Okay, I think we might have time for one more question at the back.
Thank you. I was wondering whether you've observed the impact of patients having taken, in your studies and looking at the brain, if you can draw a distinction between patients that have taken medication for depression and so on, and patients who have not, and whether that would have impacted the chemistry of the brain and how that might manifest itself in the studies you've done.
Yeah, that's an excellent question. And no doubt, these chemicals certainly do have an effect on transcription. So we have these brains and they come in after death and we have to go and do something called a psychiatric autopsy. And it's a very complex procedure, but what that means is our information about how long an individual has been taking a medication is dependent on how accurate that is. So that's one factor. So there's the effect of long-term use of a medication, which in depression is very common. People will be on drugs for a very long time. Then there's also the drugs at the time of death. And so that's kind of a different phenotype because that's a more acute effect. And so we do take both of these things into consideration when we're looking into how the brain is changing, how these gene programs are changing, and we factor them in. And we try to actually dissociate in some ways what are the effects of drugs and what are the effects of just the pathology. It's a very difficult question to address, but basically we need to do it with numbers. The more that we have with individuals who have drugs at the time of death versus those who are kind of naive at death, that helps us answer those questions and get at this refinement that you're asking about. These drugs have been studied for a long time. They have very known epigenetic and genic transcription changes, but those are not necessarily what we're after right now. We're not after the mechanism of action of drugs, so that's not really the focus of what we're looking in with these particular data.
Okay, I think we could sit here all day having conversations about, homecoming marches on, and we have to get you to your next events. Maybe instead of closing, I would maybe say two things. One is that the diseases that this fundamental work is trying to address are common. Alzheimer's disease, depression, and neurodevelopmental disorders. I think everyone in this room is probably touched directly or indirectly by those kinds of conditions. And they are complicated, and we have been trying to understand them for a long time. And it's really exciting to see new methods being brought to bear and kind of the emergence of kind of the tools, the statistical tools, and the ways of thinking about these data, and then, of course, the challenges of collecting these data and bringing them together that require bigger and bigger science, more and more people with different backgrounds. And I think McGill is really well situated to do amazing work in this area. We have the tradition of working in this area, but we also have a tradition of being unusually collaborative. This university knows how to work across disciplines, and I have great optimism that this is going to get us somewhere. We also have the support of people who are visionary about how hard this is and how you need to invest. You might be taking a chance. We don't know if this is going to work. Invest in the building blocks at the fundamental level, and have the patience and the vision to see how it's all going to come together. So thank you, Mr. Ludmer and Ms. Lander, for that. And thank you to Corina and Patricia for telling us a little bit about your work, and to all of you for being part of the conversation. Enjoy the next stages of homecoming, and good luck. Good luck with this great science. Thank you.